Hello, my name is AlidadeFlamingh and I'll be presenting some of my PhD dissertation research today. First off, thank you very much to the symposium coordinators and to SMBE for selecting my talk as a finalist in the Grad Student Excellence Symposium. I'd also like to thank all of my co-authors, without whom none of this research would have been possible. Today I'd like to tell you a story, and that is the story of how we sourced the origin of elephant tusks from a 16th century Portuguese shipwreck. Our story starts off on the west coast of Namibia, which has been subjected to alluvial or offshore mining for more than a century. Miners dry lay portions of the seabed with massive sand walls, like you can see in the images, and then dredge the exposed seabed layer for diamonds. And it was during a routine dredging in 2008 that the oldest known shipwreck in southern Africa, the Bom Jesus, was discovered. Excavation of the shipwreck revealed a very rich cargo, including gold coins, mostly of Spanish and Portuguese origin. Here you can see an example of a Spanish gold coin with the faces of King Ferdinand and Queen Isabella. One of these gold coins, the Portuguese 10 Cruzado coin, was used to date the shipwreck. The 10 Cruzado coin, shown here at the top left with a cross insignia, was minted in 1525, but these gold coins had a very high gold content, which led them to be hoarded rather than traded, and as a result there was a royal decree that these coins be withdrawn in the late 1530s. The time frame from minting to withdrawal therefore indicates that the ship had to have sailed and sank sometime between 1525 and the 1530s. This is consistent with historical records that report the Bom Jesus to have sunk somewhere off the coast of southern Africa in 1533. Also part of the cargo was more than 100 elephant tusks. These tusks were varying shapes and sizes, indicating that elephants were hunted indiscriminate of size or age. The discovery of these tusks led us to our first research question: where did these tusks come from? More specifically, where did these elephants live before they were hunted for their tusks? To answer this question, we looked at mitochondrial DNA. Mitochondrial DNA is matrilineally inherited, meaning that it's passed from mother to offspring. What makes mitochondrial DNA especially informative for studying elephants is that it can be used to establish the geographic origin or provenance of an elephant. This is because of the social structure of elephants, where females usually remain with their core social herd in a geographically restricted area. In other words, they live with their mother and aunts and female relatives in a matriarchal society in areas that are relatively close to where they were born. To see this graphically, take a look at this map on your screen created by Ishida and colleagues. They show that mitochondrial DNA in African elephants can be represented by 8 mitochondrial subclades, color-coded here. You can see that most of these subclades are geographically structured. By comparing our ivory DNA to DNA from elephants across Africa, we're able to determine which population of elephants have DNA sequences that are the most similar to the ivory and therefore likely represent the source of the ivory. Most herds will also share the same mitochondrial genome since they're female relatives, and we can therefore also use mitochondrial DNA to determine how many herds the tusks were from. To do this, we shotgun sequenced 17 tusks to reconstruct complete mitochondrial genomes. We also sequenced the shorter fragments for 44 tusks and amplified the D-loop region for 20th century geographically referenced samples. The second question that we asked was whether these tusks were from African forest elephants or African savanna elephants. And to answer this question, we looked at nuclear DNA. African forest elephants and African savanna elephants are two species that occur in Africa. Their geographic range is relatively disjunct, but there are geographic regions where both these elephants occur. However, knowing the species of elephant is still informative. For example, if we determine that the tusks are from African forest elephants, we can exclude regions in the south of Africa, since forest elephants have never been reported from those regions. To determine the species of elephant, we looked at 3 nuclear genes that contain single nucleotide polymorphisms, or SNPs, that are characteristic of either forest or savanna elephants. We also wanted to know what type of habitat these elephants were living in, and we used isotope analysis to answer that question. We analyzed stable carbon and nitrogen isotopes for 97 tusks. African elephants are mixed feeders, consuming a variety of plants including trees, grass, bark, and fruit. Stable carbon isotope ratios measured in the ivory originate from the plant foods that the elephant is consuming, which in turn indicates their habitat. African elephants living in dense forests, as you can see in the video, consume mostly C3 plants. Elephants living in more open shrub or grassland environments, like the bottom image, consume a mixture of C3 shrubs and trees and C4 grasses. Variation in nitrogen isotopes, on the other hand, is driven primarily by nitrogen cycling in the soil, which is strongly influenced by moisture availability. If the elephants from the shipwreck cargo originated from a range of diverse habitats with different rainfall regimes, we would expect to see a similarly large range in the stable nitrogen values of the shipwreck ivory. Finally, we integrate paleogenomic, archaeological, and historical methods to discuss our findings in light of 16th century maritime trade, and we also highlight past ecology and conservation implications by comparing these ancient populations from which the tusks were to those currently living in Africa. Because the shipwreck ivory contained ancient DNA, we wanted to maximize the likelihood of getting authentic, uncontaminated DNA, and we therefore carried out all our molecular work in the Mali Ancient DNA Lab at the Karl Roos Institute for Genomic Biology. We also used other methods to ensure DNA authenticity, for example, always having extraction and PCR control negatives. Also, DNA degrades in a way that has been well characterized, and we established authenticity of our ancient samples by determining that the DNA that we extracted did in fact have the damage patterns that we would expect from ancient samples. So, what did we find? We found that the ivory on the Bom Jesus derived from forest elephants in West Africa. Nuclear DNA SNPs were consistently diagnostic of forest and never savanna elephants, and mitochondrial DNA matched elephants reported from West Africa, and I'll tell you more about that. We analyzed our mitochondrial DNA by constructing a median joining network. In this network, each of the colored circles represent the 8 mitochondrial subclades that I had mentioned previously and that you can also see here in the geographic distribution. On the network, ivory is shown in ivory color, and the size of the circles represent the number of individuals that share that specific haplotype. Crosshatches indicate mutational differences between haplotypes, and the way that you can interpret this network is that circles that are close together are genetically more similar. We find that the ivory matched 2 of these previously reported subclades, namely the Western and West Central subclade. We can see in this distributional map that the Western subclade has only been found in West Africa, but the West Central subclade occurs in both West and Central Africa, and we therefore focused on analyzing published sequences for these 2 subclades. We wanted to see whether the ivory samples that form part of the West Central subclade match sequences that have been reported to originate from West Africa or from Central Africa. We again constructed a median joining network, but now only showing ivory and published sequences from the West Central and Western subclades. Haplotypes are geographically color-coded. All ivory samples matched haplotypes where at least one individual was from West Africa, and never matched a haplotype that has only been recorded in Central Africa. Together with the subclade analysis, this suggests that elephants were from West Africa. As a final supporting analysis, our mitogenome results are congruent with the tusk being from West Africa. The ivory mitogenomes clustered with an individual from Sierra Leone and an individual from Côte d'Ivoire or the Ivory Coast, and both of these are countries in West Africa. When we looked at the isotope data, we found something quite surprising. The stable carbon and nitrogen values of the shipwreck ivory cluster most closely with modern elephants from environments with a mosaic vegetation encompassing both shrubs and wooded savanna, where elephants consume substantial portions of C4 grasses. The broad range in isotopes suggests that the ivory was sourced from elephants from different habitats with different rainfall regimes and vegetation, although not from deep forests or arid environments. In this figure, the orange open icons represent elephants in drier open savanna habitats. Green open icons represent closed canopy forest habitats. Black icons represent elephants from mesic shrub and wooded savanna environments. The shipwreck ivory, indicated in black filled circles, cluster with these. Once again, this broad range of values for the shipwreck ivory suggests sourcing from multiple locations with different rainfall and vegetation patterns. So what does this tell us about the sourcing of ivory from West Africa to Portugal? Well, as you've just heard, the isotope data indicate that these historic elephants lived in mosaic vegetation. In West Africa, an example of such habitat would be the areas that surround the Ghanaian forest block. One particular area of interest is the Dahomey Gap, which is adjacent to one of the largest Portuguese trading posts during that time, called the Almina. Ivory would often be stockpiled in Lisbon before being loaded and shipped to India. Our isotope data that indicate that these elephants lived in diverse habitat, together with our genetic data that indicate that these elephants were from at least 17 different herds, supports this understanding of historical maritime trade, where ivory from different locations within West Africa were consolidated before being shipped out. These results suggest that Portuguese trade networks in West Africa in the 16th century relied on communities who hunted elephants and traded ivory through networks bordering the Ghanaian forest block, and that there was an extensive West African trade network at that time. Our study also adds to the understanding of historical elephant ecology and highlights some conservation implications. The current distributional range of forest elephants include habitats both in tropical forests and nearby savannas. Before our analysis, the recent distribution of forest elephants outside of tropical forests could be attributed to the decimation of savanna elephants in West Africa in the 19th and 20th centuries. Our combined genetic and isotope results instead indicate that the utilization of savanna habitats by forest elephants in West Africa preceded the removal of savanna elephants and dates back to at least the 16th century. Also, among the mitochondrial haplotypes identified from the 16th century tusks, only 4 have been reported among contemporary populations. This is likely reflecting the impact of the ivory trade and reduction of historic elephant range by 93% in West Africa. This drastic decrease in genetic diversity of contemporary populations highlights the urgent need for conservation of the remaining forest elephant populations in West Africa. Our data add substantially to the small body of isotopic and genetic information available for West African elephants, with the potential to aid in the sourcing of confiscated illegal ivory. We analyzed the largest archaeological cargo of African ivory ever found, and our analysis framework can be used to study the vast collections of historic and archaeological ivories in museums across the globe. Analyzing historical and archaeological ivories affords a window into the human-animal relationships across thousands of years and can reveal the formative and changing patterns of exchange between people who lived oceans apart. With that, I'd like to thank my collaborators and co-authors and other people that have sanctioned and funded this project. And thank you very much for listening. Thanks.